Good afternoon, everyone, and welcome to our webinar. My name is Jessica, and on behalf of Oxford Global, I'd like to thank you all for joining us today. Today's webinar is on the subject of advancements in oncology spatial research and will be moderated by Ioannis Winfield, Marketing Director of Nanostring. Due to speaker time zones, we are unable to conduct a live Q&A following on from the webinar. However, please do send your questions through via the chat option at the bottom of your menu display, and the speakers will reply post-webinar via email. The full recording of the webinar will be made available to download on our secured website, and the details will be sent to you after the session. If you need any assistance during the webinar, please do let us know via the chat option, and a member of the Oxford Global team would be happy to assist you. Without any more delays, I would like to now get started with the webinar. Ioannis, over to you.
Thank you for joining our webinar today on the advancements in spatial genomics research. My name is Ioannis Winfield. I am the Marketing Director at Nanostring in EMEA. Today's webinar will cover 3 short presentations, slide-based, and will be followed by a presentation of a poster. If you would like a copy of this poster, you can contact me at awinfield@nanostring.com. You can see the address over there, and I will send it to you. For now, I want to introduce you to the presentations and to the presenters for our program today. The first presenter is Professor Christina Curtis, Associate Professor of Oncology and Genetics at Stanford University in California. The title you can see on the slide is Spatial Characterization of the Tumor Immune Microenvironment Through Therapy in Breast Cancer. The work presented is addressing the critical need to identify biomarkers of treatment response in early stage HER2-positive breast cancers. The Stanford team used Nanostring Genomics Digital Spatial Profiler to quantify 38 tumor and immune proteins across multiple pan-CK enriched regions from HER2-positive breast cancers after different treatments. Important biological insights into the role of innate immune marker, ER status, and PAM50 subtypes on treatment response are presented. Our second presenter, Professor Ioannis Vlachos, from Beth, Israel, Deaconess Medical Center in Boston, Massachusetts, is presenting on spatial transcriptomics and single-cell integration for in-depth localized tissue interrogation. As you already have understood from the title, the talk will cover the method to integrate spatial information with single nucleus sequencing. The biology here is focused on the characterization of lung tissue samples from COVID patients, but the work carried out with the whole transcriptome assay on Genomics DSP is very much transferable because it's really focusing on the pathways associated to immune response and the tissue damage and repair, which would be of interest for any oncologists out there. Our third presenter is Dr. Arutha Kulasinghe from Queensland University of Technology. The title of the presentation you can see is Highly Multiplex Digital Spatial Profiling of the Tumor Microenvironment of Head and Neck Squamous Cell Carcinoma Patients Treated with Immune Checkpoint Therapy. In this study, the study actually, to our knowledge, it really represents the first spatial analysis of this tumor type and reveals that markers involved with immune cells were actually not predictive of outcome to the ICI therapy. So very interesting results. As I was saying earlier, the final part of today's session is actually a narration, a presentation of a poster. Dr. Patrick Danaher from Nanostring will share very briefly in less than 5 minutes the convolution methodology for the integration of single-cell RNA sequencing data in the tissue spatial context. If you would like to see this poster, receive the PDF, please email me at awinfield@nanostring.com. Without further delay, I would like to now leave the word and the floor to Professor Christina Curtis.
Great, so I'm really pleased to share some of the recent work that we've done using the Nanostring Genomics platform to understand how the tumor immune microenvironment changes through therapy, particularly in the context of HER2-positive breast cancer. So we've had a long-standing interest in understanding how tumors evolve through therapy. And in this particular context, we felt that a study of the changes in the tumor microenvironment during preoperative or neoadjuvant therapy would be particularly informative. And we sought to address this question in HER2-positive disease, where routinely speaking, patients are treated with standard of care neoadjuvant therapy. In this case, we turned to a clinical trial cohort where we were able to obtain pre-treatment baseline tissue biopsies prior to the administration of any therapy. Subsequently, these patients underwent one cycle of HER2-targeted therapy with either trastuzumab and or lapatinib, and they were then, at that point, obtained a second on-treatment run-in biopsy. And finally, these patients went on to obtain 6 cycles of HER2-targeted therapy prior to surgery to determine whether they had achieved a pathologic complete response or not. And so the neoadjuvant setting really provides a rich opportunity to obtain high quality tissue. In this case, we focused on profiling formalin-fixed paraffin-embedded material from some 56 patients split into a discovery and validation cohort from the TRIO-B07 clinical trial. And using the Nanostring Genomics DSP platform, we used a phenotypic enrichment strategy where we profiled a minimum of 4 different regions of interest across each time point for each patient. And using pan-cytokeratin-based enrichment strategy, we were able to specifically enrich for tumor-enriched regions. And this has tremendous value as we think about profiling relatively infiltrated breast tumor cells, but also allows us to go after the immune population that's particularly infiltrated the tumor. And separately, we can obtain the inverted mask area, focusing in on the tumor microenvironment region by taking the inversion of this. And so we profiled a total of 43 analytes per section. And to give you a sense for these data, what I'm showing here is individual patients labeled along the x-axis, where we can see whether they achieved a pathologic complete response in red, or if they didn't. We've also indicated their ER and PAM50 receptor status. And then shown along the y-axis, each dot represents a region of interest, where we're showing the protein expression levels for HER2 in blue and CD45 in red. And we can, of course, look at these ROIs and the extent of heterogeneity across each sample. So just looking at the baseline time point, we actually don't see really dramatic differences between these markers. And that was, in fact, true of virtually all markers we examined. In contrast, when we compare the pre-treatment time point to the on-treatment time point, looking at all tumor markers, we see a pretty dramatic increase in the extent of heterogeneity, with HER2, the target of these therapeutic agents, being the most dramatic, but numerous other tumor markers also increased. And somewhat surprisingly, we also see a dramatic increase in the heterogeneity of immune markers from the pre-treatment to on-treatment time point. So when we look at what's taking place overall in these individual tumors, what we can see is, in fact, by looking at a volcano plot where we're showing the change in expression in the pan-cytokeratin enriched regions of the run-in versus baseline time point, and then plot the negative log 10 FDR adjusted p-value, what we can see is that a variety of tumor markers, in particular HER2, but all of its downstream targets, are really dramatically decreased on treatment. And in fact, we see an enrichment and upregulation in a variety of immune markers. And just looking at a correlation plot, we see that, in fact, many of the tumor markers are very tightly correlated and anti-correlated with the immune markers. So motivated by the fact that we saw pretty dramatic differences, we then sought to stratify the discovery cohort by responders, those that achieved a path-CR and those that did not. And what you can appreciate from this plot is that really the vast majority of the signal is being driven by the responders. Again, we see that dramatic reduction in HER2 signaling by the on-treatment time point, and this is just after one cycle of targeted therapy, and a really dramatic increase in immune markers. And in contrast, this is simply really not very evident in the non-responders. And so we were also interested in to what extent this was reflected in bulk gene expression data. And what you can see here using matched patient samples is, in fact, that the signal is really dramatically attenuated. There is some signal in the PCR cases, but it is to a much lower level. And presumably this reflects the attenuation due to the admixed population, but also keep in mind that we had performed a pan-CK enrichment to really focus in on that population using DSP, and that's one of the great values of this approach. So I also want to just turn to the pan-cytokeratin adjacent regions where we can really profile the tumor immune microenvironment that surrounds the tumor, and to point out that using this phenotypic enrichment strategy, we can actually now ask which immune markers are enriched in the infiltrated tumor, so higher in the pan-cytokeratin enriched regions, versus those that are instead perhaps higher in the surrounding tumor excluded regions. And what we'll see here is that there's a number of immune suppressive markers, PD-L1, IDO1, that are increased in the pan-cytokeratin enriched regions. And in fact, we see evidence for an enrichment of T-cell markers that seem to be excluded from the tumor cell. So we can actually ask how these patterns change, not just at the pre-treatment time point, but on treatment and by the time of surgery. And what we can appreciate from this is actually that there seems to be an influx of T-cells, and this trend towards exclusion decreases over time. And so collectively, by using DSP, we're really able to address somewhat unprecedented questions in the context of the temporal changes in the tumor immune microenvironment through therapy. And so with that, I'd like to thank the many people who have contributed to this work, in particular Catherine McNamara, who read it, and it was really a very rich collaboration with our colleagues at Nanostring.
Before we start, I wanted to mention how excited I am to be part of this e-meeting with so many amazing colleagues sharing their work. I wanted to, first of all, to thank our hosts for bringing everybody together. My group and I are part of the Beth Israel Deaconess Medical Center, Harvard Medical School, and the Broad Institute. And in the hopefully 10 minutes, I will try to share our experience from the integration of spatial transcriptomics with single-cell technologies, some of the challenges, but also the tremendous transformative potential of combining these two modalities. So our group focuses on what we call whole genome and beyond approach. We're trying to extract from a single patient sample the maximum amount of information and integrate it for basic translational and precision medicine applications. However, the advent of two new technologies really reshaped our research program. Of course, single-cell techniques, and this is the traditional single-cell smoothie figure, where enable us to go from bulk RNA-seq smoothies to single fruit experiments. And of course, location. Spatial transcriptomics enable us to put, expression on the map, understand how localization, tissue architecture, and morphology can affect cellular transcriptional programs. And of course, the important part is that these two technologies are exceptionally complementary. From single cell, we get the missing granularity from the bulk techniques, and we can go beyond the literature. We can get our cell clusters and cell types on a hypothesis-free fashion and continue from there. What is also interesting is that even though single cell techniques are rather new, I mean, if we take the history of the world as a reference, there are plenty of data already and methods available for folks to work with. Spatial transcriptomics are the cool new kid on the block and enable us to add architecture and create expression maps, localization, morphology, and understand how structure affects function. So we are utilizing both as a single modality in our work, and we're trying to apply them on a different set of research questions. For the remaining 8 minutes, I will try to provide some high-level strategies and 30,000-foot overview, but also some of those nitty-gritty decisions that can really affect downstream outcomes. I will use as a vehicle for the rest of the presentation a data set on liver we're currently working on. So very briefly, we have human core liver biopsies, and we have the frozen portion where we're subjecting it to SNP-seq, and we have FFPE, which are subjected to the very cool whole transcriptome atlas on the Nanostring Genomics on the DSP platform, where we get 20,000 genes on the ROIs we select. This is an amazing new assay. So in our group, we do not have only data-centric labs as ours, but we have also liver specialists, we have experienced pathologists, and in a collaboration with Nanostring scientists, we were able to capture and carve out really informative, really cool ROIs that capture specific morphology features, zonation characteristics, and other liver features we wanted to analyze further. We aim for 16 ROIs per subject, and what is quite interesting, we put 2 subjects per slide for 2 main reasons. One is convenience, of course, and the second is also to be able to assess batch effects and slide-to-slide variation. I will talk a little bit about that because this is something we are asked a lot. For liver specifically, more than any other tissue we worked on, and that could be potentially how the samples were prepared or for reasons unknown to us, we see 2 important effects happening. One is we see clear segregation from slide to slide. So this is a plot showing 4 patients, 2 on one slide and 2 on the other, and they clearly segregate based on their gene expression per slide. But even if we zoom in to just 1 slide, we see them segregating again in a smaller effect size per patient. Of course, this can be patient-to-patient variation and could be biological, but its effect size is larger than the morphology and zonation we would like, for instance, to examine. So Jared, a brilliant computational biologist from our group, of course, did the first obvious thing. He took, so here we plot only zone 1, 2, 3, with brain orange and purple, and you see the segregation. And he modeled out, slide, and he modeled out the patient effect. And you see, for instance, for the patient, you get a more homogeneous representation, but however, you lose a signal of zonation. So he devised a method where you rescue the biological signal from known and unknown biases, and you continue working with the residuals of that model. So on the bottom, this is not new in computational biology. This has been established by Jeff Leake's group for many years now. And in this adaptation of this approach, we see here the outcome. So these are the BSSR residuals. We're not working on expression space anymore. This is the residual space. We can see that not only similar colored ROIs huddled together, but we keep this natural progression from green, orange, and to purple, which is zone 1, 2, and 3. So this is definitely very exciting, and we wanted to check, how does this affect the biological signal? So we see here 2 heat maps. These are genes in the literature that are relevant to zonation in the liver, and we see, for instance, that in the BSSR, these are the residuals. These are the normalized counts we're using as an input. Upper 3rd quartile normalized counts from a lot of help from the Nanostring specialists. So we can see the progression from central hepatocytes, zone 1 to 3. These are markers for portal hepatocytes. Again, zone 1 to 3, very gradual decrease. Periportal pericentral endothelial cells 1 to 3, and periportal. If you go to the original, you see, for instance, a mixed signal. And that makes us, first of all, very happy that we are able to salvage this very refined biological signal from, for instance, how the slides were prepared. And we use this residual space to all our downstream analysis. Now this is where the fun part starts, right? We have single cell experiments. And from the single cell, we don't need to use the markers that I just showed that are from the literature. We can find the actual clusters in the pathology that we're studying. We can find subclusters. We can find markers annotate them, do cellular trajectories, and of course, we can do also cell signaling networks to understand which cells are talking to each other. But having spatial data really can transform what we can do, because from the cell types that we have identified in the markers, we can deconvolve and register them on the ROIs. And that can enable us not only to compare the different zones and different ROIs using gene expression, but also cellular profiles, and can inform. That's why the arrows have two edges, and that can also inform the single cell. We can compare zones in the spatial and check which cells are responsible in the single cell. We can, for instance, answer the fundamental question: I have this very interesting macrophage population in my single cell. Are those macrophages in a specific region all together? Are they scattered across the tissue, or are they in specific subregions? And if yes, what are those regions and what other cells are there? And what is the most transformative aspect, at least in my opinion, is that we did before, and as everybody does, right? You have your single cell and you do your signaling network, but you do not know whether those cells are actually adjacent to each other. You can create spatially aware signaling where you can keep the populations. You get your ligands and receptors from the single cell, and you create your network within the ROI. So another thing that we tend to forget, and I wanted to share this with you, is that we focus a lot on the expression space. Everybody does, but we forget that these are images and very informative, where we can, do image analysis, count the cells, apply AI, connect gene expression with cellular morphology and cell counts. And we can also do, image analysis techniques. So let me give you a simple example. So if we see the zonation in liver, it creates two-dimensional vectors that have, different orientation and different lengths. But because we also get the coordinate matrices for the ROIs in the tissue, we can apply scale and rotation invariant modeling. And that way, we can, for instance, model gene expression as, for instance, distances from specific landmarks, regardless of scale and orientation. There are a lot of things to do on the image plus expression space. I started with this slide and I wanted to close because having both for us has been a completely game changer. We get our cell types and our subtypes from the single cell. We get our very specific for our pathology markers. We have seen a very big difference when using, for instance, atlases from healthy tissues versus atlases we create for our specific pathology and applying them on the spatial. We get our signaling relations and our deconvolution reference. And then on the spatial, we get, the architecture, the map. We can bound cell populations in space, and we can do spatially aware signaling networks. So closing, I wanted to, first of all, to mention that this project that I briefly discussed is part of an extensive effort in the IDMC. This is a group effort among 8 PIs. I wanted to shout out to my colleagues, John G., Stefan, Wynn, Yuri, Linus, Gordon, and Jonathan, who are part of this amazing initiative, and of course our lab members who are helping with the analysis. Jared is spearheading this effort, but definitely nothing would have been made possible, if we didn't have Nanostring support to bring us close to this technology, provide us access to experienced engineers and scientists, and help us and guide us through this process to really maximize the outcomes and the utility and the potential of our research. So please reach out to us, find us online, find us on Twitter. We have many fantastic things cooking in this space.
Hello, everyone. So My name is Arutha Kulasinghe. I'm an NHMRC Early Career Research Fellow from the Queensland University of Technology, and I'll be talking about some of my spatial proteomics work in head and neck cancer patients that received immune checkpoint therapy. So this work started off, so my background is in looking at circulating tumor cells. So these are transient metastatic cells or metastatic precursor cells that are found in the blood of solid tumor patients. They're typically found as single cells or cell clusters, and I was really interested in these rare cell populations in the blood. And obviously they go on to form distant metastases in the lungs, liver, and brain. So we used a number of approaches to capture these circulating tumor cells, as described in the slide here, including positive selection. So we used FDA-approved EpCAM enrichment technology, as well as negative depletion to capture these rare cells. We also looked at filtration-based devices and microfluidic devices to capture these rare cells from a blood sample. And these are some of the technologies we looked at, and we looked at the plasma as well for clinically actionable mutations. So when we were characterizing these circulating tumor cells and phenotyping them, we found that PD-L1 was frequently expressed in these head and neck samples, and that was really interesting to us, and we wanted to look at it in a bit more depth. And we also performed ex vivo culture in a number of these samples. So this is one of the key slides where we found that in circulating tumor cells, PD-L1 was frequently expressed, and it was expressed in head and neck and non-small cell lung cancer. And this is compared to a number of cell lines. Obviously, PD-L1 is dynamically expressed in the tumor, so we wanted to find a more realistic or real-time snapshot of the PD-L1 expression in CTCs, and we thought that this might be a way in which to do that. And this is just some survival metrics at the bottom right-hand corner, showing that patients with CTCs had a poorer overall survival in head and neck and non-small cell lung cancer. So that's, I think, the time when Jim Allison's work came out, and the work in CTLA4 and the science papers that came out showed that in a certain subset of patients, they did really well with immune checkpoint therapy. And this was really driven by melanoma, but head and neck and non-small cell lung cancer, as you can see, were were a subset of patients that benefited from this therapy. So because we were working in this space, I really wanted to go back and look at the tumor of these patients and see what was going on, not just at the PD-L1 expression level or PD-1 levels, but other tumor microenvironment cells as well. So there were a number of biomarkers in this space, and TMB, or tumor mutation burden, was one of them. So on the scale here, you can see that head and neck and non-small cell lung cancer sit in the sort of midpoint of this graph, and high TMB for head and neck is about 10.3 megabases per mutations per megabase or more, and then this is being refined at the moment, and it's sort of being led by melanoma and the data in the first line setting there. For head and neck, it is given in the metastatic setting, but there is it is now being looked at in recurrent head and neck patients in the first line setting. So this comes on to the tumor microenvironment work that I'll be talking about in this talk in a greater depth, and actually looking at this spatially and getting that level of depth of information, not just expression data at the global scale, but in looking at different regions within the tumor microenvironment was really, really key to moving this work forward. And we were able to do that using the Nanostring digital spatial profiler in a pilot study, and we're expanding this work at the moment. So really looking at the microenvironment versus the tumor stroma, and comparing it to normal regions to see what was driving these cells. So this is some of the therapies that are given for head and neck, or the progression of it over time. And you could see that in the mid to early 2000s, when PD-L1 was found to be overexpressed in head and neck tissues, and then there was iterations of Pembro and Nivo that came out in the last couple of years, and then where the field is heading now with a number of immunotherapies being developed in this space. So this is the technology we use to profile a number of head and neck tissue samples from patients that received IO therapy. They were recruited to Checkmate studies, and it was a preliminary study. I'm sure the audience is aware of this technology. So they're antibodies. So we looked at the proteins initially, and now we're doing the RNA. So we looked at the protein target. So we were interested in profiling these tumors using the 40-plex immuno-oncology panel from Nanostring, where the antibodies bind to a UV photocleavable oligo, and when regions of interest are chosen, the signal is liberated, and that can be counted. And there's a number of different data normalizations. So I think this is a key slide for a lot of this work. I think it speaks to a lot of this new technology, and then single cell sequencing, and then the data normalization is becoming increasingly challenging. But I think there are ways in which we can better assess our data. So there are a number of different ways this data was normalized. So ERCC normalization, nucleus normalization, area normalization, and a number of housekeepers include, and signal to noise normalization for each data set. So we were able to measure across the ROIs and within the ROIs if there were tumor or stroma. So these are a number of, just a snapshot of what we do. We get an H&E section. This is assessed by the pathologist to demarcate the tumor and normal region. We tried to do whole exome sequencing on these tumors, but there were limitations with how much DNA was available from them. From one of them, we were able to get enough DNA to do a whole exome, and that patient had a TMB of 5.7 mutations per megabase. So this is your typical H&E stain on the top, and then in the middle, what we can see is a visualization marker stained by the DSP, and stained and imaged by the DSP for pancytokeratin, CD3, CD8, and DAPI, where we would then compare that staining to the H&E and then choose regions of interest. So you can see a number of regions of interest. For these, it was 12 regions of interest per tissue section. And then on the corresponding slide below, those are the regions of interest at a higher magnification. So we did this for 8 tumors, 8 HNSCC, so head and neck squamous cell carcinoma tumors. So this is a high resolution image of one of them. So you can see that this has been stained with pancytokeratin, CD3, CD8, and DAPI. And then these 12 regions of interest are selected, and then those are profiled at 40-plex. So the visualization markers are key to choose what you want to profile. So ideally, you'd want to do more if you could. So I think we are looking at doing 24 and expanding this out even more. So this is just some of the data from a head and neck patient that was T4N0N0, left mandible P16 negative, so HPV negative. This patient had multiple rounds of chemoradiation and surgery and was given immunotherapy, so nivolumab in the metastatic setting. So we had the diagnostic biopsy tissue, we profiled it using the DSP, looked at 12 regions of interest across the tumor, the normal, the stroma. And this is just a snapshot of at the bottom, the graph just shows the variation in individual markers per region of interest. So we can see whether tumor-rich region is high in cytokeratin, or just to get to understand whether the assay worked or not initially before we go into the multi-marker work. And the panel of antibodies we looked at are on the right-hand side here. So there's a number of controls, a number of housekeepers, and the rest of the markers span immune activation, immune cell profiling, IO drug targets. So a number of novel markers as well in the STING, VISTA, IDO1, which are really interesting markers, I think, in the IO space. And some pan-tumor markers as well. And we were able to do this across multiple tissue sections and generate heat maps essentially for each tissue section. So where we're able to measure a tumor signature, an immune signature, and then infiltrating edge or mixed signature where there is overlap between tumor and immune regions together, and just to get an idea of what cells are there and then the certain marker expression levels. And we're able to do this from about 20 microns all the way up to 400 microns. So it depends on what your region of interest is and what you're looking at, whether it's in the global level or you want to go down into the sort of the 10 cells or profile 20 cells at a time. And this is just a snapshot of what we're able to do. So this is the 40 markers measured across this one tumor, just showing different expression levels, just to see what sort of data we're able to get. And on the right, what we see is the variation in three markers which are being looked at in the IO space, so VISTA, STING, and IDO1. And you can see the variation across three head and neck tissues for these three markers. So they're involved in immune regulation, and we're trying to look at this in greater depth. So not just the PD-1, PD-L1, but what about the other markers? And then spatially, where is VISTA expressed? Where is STING expressed? Where is IDO1 expressed? And how does that associate with disease progression? And this is another snapshot of the variation across those 8 tumors for the tumor, immune, and mixed ROIs, and the variation across those markers going across the chart there. And in this slide, we're able to compare tumor regions versus immune regions and see what sort of cells are expressed there spatially, what cells are within the tumor. It's not just about the CD8 cells within the tumor, but I think what other cells are there. I think some of David Rim's work showed that CD68 cells in the tumor were predictive in melanoma. So I think, not just looking at the CD8 cells, but what other cells are brought in, the NK cells, what are the FOXP3 cells could be really interesting. So we're able to compare different regions of interest and see what sort of markers are expressed, what sort of immune cells are recruited into the tumor or excluded from the tumor. And in the bottom, we're able to measure the infiltrating edge versus an immune-rich area as well. So the same sort of analysis, just giving us a snapshot of the signal within those areas. So validation is always important, and this was quite challenging for us because we were looking at 50 or 60 immuno-oncology markers spatially and how we could then validate this data. So we looked at using a smaller multiplex IHC panel. So in this case, 4 markers, but we built it up to 6 markers at the moment, using the PerkinElmer Opal staining kits, so CK, CD8, PD-L1, and DAPI were done on similar regions of interest. So in the top, you can see an H&E section demarcating the tumor, and then that was profiled on the Nanostring and the Opal systems and assessed for similar regions of interest to see how the expression levels varied. And we were able to find that both systems were quite concordant with what they were the staining across both systems. And this was reaffirming to us, and we're doing this in greater depth at the moment. So this is some analysis between the total set of samples done. So the patient clinical pathological findings are on the left-hand side. So 2 patients received Nivo and 5 received Pembro in the metastatic setting. And we were able to compare the marker data to survival outcomes. So whether they were progressors, whether they were non-progressors, NP demarcates non-progressors, and PD demarcates progressive disease. And what was really interesting from this initial pilot data set was that CD8 cells weren't predictive of outcome. And this sort of goes against the current literature, but I think this was interesting because spatially we could assess this. And what's interesting was on this next slide, we could see that there were a number of markers that were predictive of progressive disease. So CD44, STING, CD66, phospho-AKT, and PTEN. So now we're looking at these markers at a greater depth and really trying to understand spatially where they are and what other cells are recruited within these regions. And PD1 and PD-L1 at the top of this graph here, and granzyme B. So this is just some preliminary lung cancer data where we did a tumor microarray, so 92 cases with tumor and matched normal, and looked at it on a 60-plex IO panel, again, protein. And what we wanted to do was use the masking function. So this is where you're able to take the pathologist's demarcation of the tumor and normal regions, and then you're able to draw within it. So in this, in the bottom right-hand corner, you can see that you can make a tumor mask, and you can liberate the signal of that. And when you do the inverse, you get the tumor microenvironment signal, which is, you get a total signal per core for the tumor and the microenvironment data. And this is what we're trying to attempt with the lung TMA. And this data is really, really new. So I'm really excited about this data. And just a quick snapshot of what we're able to do when we're able to compare the stroma versus the healthy cell. Each core had a matched normal tissue as well. And we're able to see the number of markers that are highly expressed, which are significantly found to be present between the healthy and tumor versus the stroma and tumor as well. And VISTA comes up again when you compare the tumor versus the stroma, CD44, CD45. So there are a number of markers here that are highly significant that we're trying to look at in a greater depth. And at the bottom, that's just a heat map comparing the tumor, the stroma, and the matched paired normal. So this is really preliminary data, but it's really exciting that we're able to do this from a tumor microarray. And we're hoping to really use that geomasking function to get two signals or two signatures per core, and then we'd be able to measure it across a number of TMAs. And I think this is really powerful. In this case, this was in non-small cell lung cancer. So in summary, multispectral spatial proteomic profiling characterizes the tumor microenvironment in HNSCC patients treated with IO. CD8 T cells were not found to be predictive outcome to immune checkpoint therapy. There were a number of other markers, CD4, CD68, CD45, 44, and 66B, were found to associate with progressive disease in a preliminary cohort of patients. And in one patient, there was a low to intermediate TMB for that tissue section. And so the DSP data revealed an increased PD1 and low PD-L1 in the tumor ROIs. So comprehensive analysis of the TMB and spatial TME, so tumor microenvironment, may in combination predict outcome in the future. And these applications, I think, can be extended into CTC analysis and organoid cultures if you can get if you can get those cells on slides, and then you'd be able to profile them. So I think that's a really novel angle that I'm looking at as well. With that, I'd like to thank you for listening to the talk and to acknowledge the funding agencies that contributed to this study, as well as the patients that gave their valuable samples. And I'd like to thank the team in Brisbane that assisted with this work as well. Thank you.
Hi there, thank you for joining. I'm Patrick Danaher. I'm a principal biostatistician here at Nanostring, and I'm going to be talking about updating immune cell deconvolution for the spatial genomics era. In very brief, our first two panels here are showing the platform and the Cancer Transcriptome Atlas panel, the 1800 gene RNA panel we're using in this experiment. I'll leave you to read about those. So the essence of what we're doing is in this cartoon on the left here. We've got a deconvolution algorithm, and I'll show you how this works. So you would begin with a tissue sample, and on the left branch, you would collect Genomics RNA platform data from this sample. On the right branch, you would use single-cell RNA-seq or publicly available data to derive a matrix of expected expression profiles for all the cell types in your study. So this is the expected expression profile of a T cell, of a B cell, of a fibroblast. Then with those two inputs, the genomics data and this cell profile matrix, you put them into the algorithm and get out a spatially localized estimate of all the abundances of all these cell types in your study. So this is T cell abundance, B cell abundance, fibroblast abundance within each region of the tissue. All I will say about the algorithm is that deconvolution, it's pretty rich literature right now. Our major contribution is to replace the least squares penalty at the heart of most deconvolution problems with a log linear penalty, sort of akin to log normal regression, and that has quite a few benefits to it. At the bottom here, we've validated our approach. We've compared in adjacent slides from the same tumor our estimated cell type abundances versus expression of canonical protein markers, and we've found very good correlation. This appears to be working quite well, even in the challenging data posed by small regions within FFPE embedded tumors. But I really want to spend most of my time on the application of this method, just to show the rich data you can get from localized cell type abundance measurements across a tissue. So on the top here, you can see a non-small cell lung cancer tissue. We've taken a grid of 191 regions across this tissue. And then within each one of these square regions, we haven't just profiled that little square, we've actually collected data from it in two different ways. First, from just the tumor cells, or the cells that are expressing pan-cytokeratin, and then second, from everything that's left, so the stroma or the microenvironment. So we've got 191 regions, and in each region, we've got these paired measurements of tumor expression and stroma expression. And the importance of this segmented approach is shown on the right here. There are some genes which, CD45, EpCAM are very specific to each of these segments, to the microenvironment, to the stroma. And then these other genes are examples of genes that, you really can't interpret in mixed tumor and stroma unless you collect them separately. If Ki67 is high in tumor, that's a bad thing. If it's high in the microenvironment, that's probably a good thing. It's a totally different interpretation. You really can't disambiguate them unless you collect those data points separately. So the results of our deconvolution algorithm in this tumor are shown in the middle here. The little wheel there gives a color key for all the different cell types we're measuring. Just to make it simple, we can look at one cell type at a time. So looking on the right now, you can see these dense B cell enriched regions. B cells, not only do they cluster on one side of the tumor, but they're actually very condensed within just a few regions of the left side of the tumor. T cells, or CD8 T cells in contrast, they tend to be enriched around the regions where the B cells are, but they're diffusing farther out through the tumor, almost in a gradient away from the B cells. So this is kind of a model where you might have these B cell enriched tertiary lymphoid structures sending out instructions, and the CD8 T cells are invading out from those regions. We see Tregs basically following the CD8s. You don't see high CD8 T cells without the inhibitory Tregs. And then macrophages have a very distinct pattern. They're spread in a low level throughout the tumor, and then they also have this pocket of enrichment in the lower right, kind of away from the lymphoid populations. So yeah, totally different pattern. We can also cluster our cell type data to come up with subtypes of the localized immune response. So here, if you look at this bar plot and heat map, showing the same data, you can see we've got a macrophage-enriched subtype, which is really dominated by these dark green macrophage bars. We've got a mixed subtype, which has T cells and macrophages and monocytes, and a tiny bit of B cells. And then there's a heavily B cell enriched subtype, which also carries a lot of T cells along with it. And then there's a T cell dominated subtype, which very small numbers of monocytes and macrophages, very, very few B cells, and primarily CD8 and CD4 T cells. If you look below, you can see where these four localized immune subtypes cluster throughout this tissue. And there are spatial trends to them. The macrophage subtype favors the right side, the B cell subtype favors the left, but they're all intermixed with each other. It's not a gross gradient. It's a pretty fine pattern we see throughout this tissue. And then finally, we can compare, a natural question is, how does the tumor respond to the localized immune infiltrate? So this heat map here is showing a correlation analysis between the 14 cell types that we measure in the microenvironment compared to the gene expression in the adjacent tumor cells. So every point in this heat map is a correlation between expression in a tumor and the local abundance of a given cell type. And there's far too much information here to display in a poster, but we can see little clusters of genes that are possibly correlated in the tumor when CD8 T cells are abundant versus when macrophages are abundant. And we could spend a whole other poster diving into these results. Anyway, thank you very much.
This brings the webinar to an end. On behalf of Oxford Global, I would like to thank everybody for joining us today. I would like to say a big thank you once again to all of our speakers who put a lot of time and effort into making sure that this webinar was interesting and valuable as possible. As mentioned at the beginning, we are unable to have a live Q&A. Thank you all for submitting your questions. We will send these across to the speakers who will be in touch shortly via email to answer. If you would like a copy of the posters shared during the webinar, a reminder that you can write directly to Agnesi at awinfield, which is WINFIELD, at nanostring.com. Nanostring will be hosting a workshop next week on the 4th of November as part of the Next Genomics virtual event. This will cover from single cell to spatial genomics in oncology, neurology, and transplantation research. If you would like further information on this or would like to register, please visit the website or contact the Oxford Global Marketing Department. All that remains to be said is to have a great day, and we look forward to welcoming you to one of our future events. Thank you.